Our next speaker, Dr. Shafeer Kalathil, Assistant Professor in Microbial Chemistry at Northumbria University, Newcastle, England. Before Northumbria, he was a Marie Curie Fellow at University of Cambridge, a postdoctoral fellow at KAUST and Japan Society for Promotion of Science Fellow at University of Tokyo. His research group at Northumbria University explores electrically active microbes to produce electricity or high-value chemicals from waste carbon. He has published more than 35 international papers in top journals, including Nature Catalysis and Nature Energy, and has 10 patents to his credit. Currently, he is a Fellow of Higher Education Academy of UK, member of Royal Society of Chemistry. Also, he is an associate editor of Frontiers in Microbiology. Now, it's my pleasure to welcome Dr. Shafeer Kalathil for the presentation on the topic Microbial Solution for Climate Change and Energy Crisis.
Thank you so much for your introduction. So now welcome to the world of microbes. So now we are facing two challenges, right? One is climate change and energy crisis. The question is whether microbes can save our planet. So my research is on a special microbes which is electrically active. That means they can produce electricity from waste. So today's talk, actually I'm talking initially how we can use bacteria or microbes to produce electricity. So in our lab, we are actually running the fan or bulb using bacteria. So we can actually expect that one day we can run the car using bacteria. So how bacteria producing electricity? Then how we can use bacteria, this kind of electrically active bacteria, to produce catalyst. So you people know that what is the function of catalyst. So how we can use bacteria to produce electrocatalyst. And finally, I'm actually mimicking the photosynthesis. You know the photosynthesis, right? How we can mimic photosynthesis using bacteria. So all the topic actually covering using microbes. So just follow me how we can use the special microbes to produce electricity. Okay. So earlier we discussed that we have lots of science branches like chemistry, physics, biology. In reality, now there is no pure science. There is no pure chemistry, no biology, no chemistry. Now only interdisciplinary. That means we are merging together all the branches to make some good, exciting science. In my case, I am using biology for my research, especially genomics, microbiology, and proteomics. And also I am heavily using chemistry, especially spectroscopy and electrochemistry. So I will talk how I am actually integrating chemistry, biology, and physics. And also I am using physics, especially microscopy, to evaluate the performance of microbes. So if you want to do some exciting science, you should have knowledge in different fields, especially the fundamental science we call chemistry, physics, and biology. So I will show how we will use this kind of knowledge to interpret microbial behavior. So if somebody is asking you what is life, this is one of the fascinating definition of life, the Nobel laureate Albert Gyogi. So if somebody asking about life, the simple definition is life is nothing but an electron looking for a place to land. That means simply the flow of electron means the life. If there is no flow of electron, we are dead. So the life is sustained by the flow of electron. So today's talk is all about electrons, how we can use electrons and how we can actually hijack electron to produce energy. So for our case, for human being, here you can see in the bottom. So how we are sustaining the life? We are inhaling oxygen, right? And releasing carbon dioxide. That means aerobic respiration. So this is the major respiration in living world, this aerobic respiration. Most of the living creature, including humans, they are actually taking oxygen, releasing carbon dioxide. But there is a second type of respiration called anaerobic respiration. You know, if you go to the sea, the deep sea, there is no oxygen. but still you can see the life. So there are some clever bacteria or microbes, they can still alive without oxygen. How they are actually doing that one? They are using some soluble electron acceptors and donors. For example, here, nitrate, sulfate or carbon dioxide they can use instead of oxygen. So this is also typical respiration. We are calling anaerobic respiration. And there is a third category, that is my topic and very recent discovery. So it was discovered a decade ago. That is extracellular electron transport. For example, if you go to the extreme deep sea, there is no soluble electron acceptor, there is no oxygen, but still you can find the microbes. That means they are actually living by eating rocks. You know the rock? Normally rock is made of iron oxide or manganese oxide. The speciality that they are not soluble, right? For example, if somebody want to inhale something, it should be soluble form. You cannot inhale rock or something. But there are some clever bacteria that can actually inhale rocks. They are just eating the rocks and living. So I will show a video. Yeah. This is a live video. We shoot it using a high-speed camera. So you can see that how bacteria eating rocks. So this is bacteria, they are swimming towards somewhere. You can see a spot there, a dark spot. This is called a rock made of manganese oxide nanocrystal. And you can see that this is a rock, a manganese oxide rock. And bacteria going and eating them. And this manganese oxide is insoluble. Okay, you can see as a rock. And eventually you can see that it is disappearing. That means manganese oxide is in +4 stage, which is insoluble form, which is bacteria eating and dumping the electrons to the manganese oxide and eventually converting manganese 4+ to manganese 2+, which is a soluble form. So we can see that the size is decreasing, right? You can visualize that one. And eventually it is disappearing. That means bacteria ate all the available manganese oxide and converted to a manganese 2+. And you can see that now bacteria is not moving. We are calling Brownian motion. That means there is no more manganese 4+. They are actually stopping. Now they cannot breathe. So their breathing means they are actually giving the electrons to the manganese oxide and finalizing manganese oxide 4+ to 2+. Normally it is very difficult to convince people that bacteria eating rocks. That is why we shoot it using a camera. This is actually a 1 week videograph we shot it to just 1 minute. We are using a high-speed camera overnight, and we can visualize what bacteria is doing. So it can actually give you a visual impact how bacteria are living using rock. Okay, this is one of the very hot topic, and it is still enigmatic. That means we don't know the real mechanism how they are doing. So that is my one of the research topics. I will show some of my papers which I published in this area. So here during my PhD time, I used this special bacteria to produce electricity. And in actually, the thing is that when you go to the microbiology textbook, you can't see any bacteria which can make electricity. So usually we are saying that a tale no one told us before. So the interesting thing that here we can use a special bacteria. You can see the microbial fuel cell. What is happening that we are actually feeding wastewater to the anode chamber of that fuel cell reactor. And in wastewater, there are many bacteria which is actually oxidizing organic compound in the wastewater. For example, glucose. You know the oxidation, right? Oxidation means release of electrons. So what is happening that we are hijacking the electrons using the electrode, and we are actually dipping the electrode to the microbial fuel cell. Bacteria oxidizing the glucose, and the electrode is taking the electron, and electron is moving to the cathode. That means there is a flow of electron, and eventually it is making electricity. So we can use this device to produce electricity from waste. So as you are young minds, maybe after 10 years, this will be one of the very hot topics. So I am giving a hint for your research. The question is that how bacteria giving electrons to the electrode is a very, very interesting question. So here we propose in our study that, unfortunately I cannot point it here. Yeah. Here, this is the bacterial inner membrane. So if you know the structure of bacteria, there is an inner membrane like a quinone pool, and there is a periplasm, and then finally an outer membrane. So bacteria making electrons here, and they are actually transporting the electron to outside through a series of protein network. This is like a bridge. They are making some bridge internally to connect externally with the electrode, and this is called extracellular electron transport. So bacteria has a special mechanism to transport electron to outside, but still there is no direct proof. So we are predicting it. Okay. So for that, to prove that one, we did some research. Yeah. So another interesting thing that this bacteria has a special appendages, that is structure. We are calling like pili type string. So can you see here? This is also a video graph. They are making a structural nanowires, like they are connecting each other. So the specialty of this wire is that they are electrically conductive, like copper wire. So what they are doing that when two bacteria coming together, they are doing communication. We are calling technically quorum sensing, microbial communication. So they are actually giving the signal each other using this electrical signal. This is called electrically active nanowires, or e-pili. So here also you can see the graphical representation how bacteria make a nanowire, which is actually highly conductive, and eventually they are communicating each other. So this discovery in 2009 selected as the best invention in 2009, because this is extremely interesting that how bacteria making conductive nanowire. So you know, if something is conductive, normally there should be a flow of electron. So we are using, actually I am using this bacteria more than a decade. So my PhD topic was on this bacteria, and still I am working on this bacteria. So this is a special bacterium which can make electricity and which can make electrically conductive nanowires. So this is one of the very interesting topic, how they are actually making electrically conductive nanowires. So if you look at the life history of the bacteria which I am using, it actually started in 1994, the discovery of the bacteria. So people usually asking me that I am working with the bacteria, so whether it is pathogenic or dangerous. So in bacteria, in the microbial world, there is classification. We are calling in American style biosafety level 1, 2, and 3. If it belongs to biosafety level 1, you don't need any precaution. So fortunately, this kind of bacteria belongs to biosafety level 1. So you can even work here without any precaution. And also, if you go outside and grab a sediment outside, just outside here, more than 60% of bacteria belong to this sediment, belong to this category, that is Geobacter sulfur-reducent. And the genome sequence actually discovered in 2003. And now it is the final thing, like the last 4 years, we know that they are actually making conductive nanowires. And in Cambridge, actually, when I joined Cambridge University, one of the bottleneck in our field that when we are actually taking the electrically active bacteria, they want a host like electrode, which is highly conductive and porous. So that was one of the bottleneck in our field. So I developed a structure in Cambridge, which is highly porous in nature, which actually helping bacteria to penetrate. You can see an in situ videograph, which is actually showing that bacteria actually penetrating through the pore size. This is a pore. So bacteria actually moving through that one and staying there and making biofilm. Biofilm is the family of bacterial, single bacteria. Okay. So they are making a biofilm inside. And why I am showing this kind of images, when you are doing research, you will use very sophisticated instruments to analyze and visualize the phenomenon. So here you can see the X-ray image, which is showing that a three-dimensional structure. Okay, you can actually visualize three-dimensionally what is really happening. And here, bacteria moving through this pore and making biofilm. And using this structure, you can see this panel, we got a record current density, like 3 milliampere per centimeter square current. So we got a current from bacteria, which is actually run a small bulb in our lab. So what we can do that when you are using bacteria, you can run a small bulb or table fan using bacteria. This is the measurement. In electrochemistry, we are calling chronoamperometry. So you can see that they are actually happily making electricity, which is actually you can run a fan or even a table lamp using bacteria, okay? So this is one of the main discovery I did during my Cambridge life and published in an American journal. Then what I did that we can actually sequence the genome, like for bacteria, usually bacteria or the human body, we have genes, right? So each gene has special function. So here, for the geobacter bacteria, there are around 5,000 genes for this bacteria. So we want to know that what gene is actually really doing the electricity production. Then only we can actually manipulate genetically manipulate. So we did genome sequencing, and we found that they are using C-type cytochrome genes, which is actually functioning as a key role to make electricity from bacteria. So if you are doing research, you need to do the fundamental insight, what is really happening inside the cell body. We found that they are really making electricity using C-type cytochrome. Cytochrome is a protein structure, which is actually helping. Because C-type cytochrome means I think you learned that heme group, right? Heme means iron. So the iron has a 2 state, +2 and +3, ferrous and ferric. So it can actually do the electron hopping. So even though you cannot grab me completely, that is fine. The thing is that if you are doing research, you need to dig deeply to understand the mechanism. So here we are actually understanding the mechanism by doing the genome sequencing, and we found that there are certain genes which is responsible for making electricity. Then very recently, I got interviewed by BBC Radio last time, whether we can use the plastic, so like this kind of plastic bottle. So one Japanese group actually discovered a bacteria which is eating the plastic bottle. The name of that bacteria called Idionella sacensis. So that means if you give this kind of bottle to a special bacteria, they will eat this plastic bottle and converting this plastic to acetic acid and ethanol. So that means we can actually address a challenge, that is plastic pollution. Eventually you are making very useful chemical. So in Cambridge University, I discovered that there are certain bacteria, if you crush this bottle to that bacteria and feeding them, using a certain pathway, they are making acetic acid and ethanol. So it got published in one top journal in Angewandte Chemie International Edition. So, and also we found that if you feed the plastic to the plastic eating bacteria, they can also make electricity. So that means you are just giving the plastic bottle to bacteria, you can get electrical current. So you can get electrical current or some chemical like ethanol or acetic acid, which is actually a breakthrough in plastic pollution research. So only drawback of this system is that the system is very slow. So now we are trying to speed up the reaction. Then if you speed up the reaction, we can just feed the plastic bottle to bacteria, and we can get either energy in the form of electricity or chemicals like ethanol, acetic acid, butyric acid, like medium chain fatty acid type thing. So this is really a cool experiment how we can use bacteria to convert plastic to the useful chemical. So here in Newcastle campus in UK, this is our building. We recently built a green building in our campus. This is our campus. This building, the specialty of this building is that all are green. That means we have a toilet, we have an active kitchen, we have a bedroom, and we have a living room, and we have a special toilet which we imported from Switzerland. The toilet actually is separating liquid and solid. That means we can separate the urine from this toilet. So what I am doing here is that once we separate the urine from this toilet, I am making electricity from urine. So I am giving urine to the bacteria, which I collected from this toilet. So this toilet will give the urine separately. I am taking that urine and give it to the microbial fuel cell reactor. And in chemistry perspective, urine is one of the good electrolytes. So if you look at that chart here, the urine chart is highly conductive because of lots of trace metals, and we have creatine, glucose, fructose, which is organic compounds, which bacteria like most. And we are actually, what I did that, I fed the urine to the microbial fuel cell. What is happening that the bacteria in the urine oxidize in the organic compounds in the urine and making electricity. And another interesting perspective that urine contains lots of nitrogen and phosphorus, which is actually a good nutrient for as like a fertilizer. So we are actually recovering nutrients from urine, and also we are getting electricity, and we will get treated water. That means what we are doing that once we pass the urine, we will get electricity, we will get nutrients like fertilizer, which I am using actually. Now we have a garden here and here. So we are using that fertilizer which we recovered from urine to garden here, and electricity we are using to run our fan and bulb inside in the kitchen. So what means we are getting every energy from our waste, from urine. That is my research. There are other people doing using kitchen waste. So anyway, this building is a green building, and from the building whatever waste is producing, eventually we are converting to energy in the form of electricity or nutrients. Now what is we are doing that using, in collaboration with Bristol University, now we can use this urine powered mobile charger. So we can actually charge your mobile using urine. So urine will be the source to charge the mobile phone battery. So at the moment we can get a power at least to power a mobile phone charger. So yeah, here you can see there is a urine inside. Inside is urine, and you can see the LED bulb is blinking, right? So the electricity actually came from the urine. So it's a demonstration to public that how we are using urine to power a bulb. So usually we are using this kind of demonstration for Science Festival to make the public attractive. So we are showing the public that what we are doing. So inside is urine and bacteria which we collected from that building, and it is making electrical current. So you can see, I don't know whether in the long distance you can see the blinking. So you can see the LED bulb is blinking using the urine collected from the toilet. And also this is the research I did when I was in Saudi Arabia. So I got an American patent. We can use electric bacteria to make some catalyst. So you know the water splitting, right? You can split the water to hydrogen and oxygen. So we are using different catalysts to split the water. So one of the catalysts is manganese oxide. So we are producing manganese oxide using bacteria to split the water to oxygen and hydrogen. So eventually you can see that this is a bacteria which is decorated with the manganese oxide, and you can use this structure to split the water. So you can split oxygen to oxygen and hydrogen. Hydrogen can be as a fuel. So this is we got a patent for this one. Then I am going to my final research which I am actually doing now, how we can use bacteria to address climate change. So if you look at the UK, because we are actually based on UK, UK is a winter country, right? But in summer, last summer in 2023, we got record heating, especially in London. 40 in London is a record heating in London. And I'm coming from Newcastle. We are fortunate, we are like 32 because it's northeast England. Anyway, the London actually recorded 40 degrees Celsius, which is very high. This is showing that we are actually facing the global warming. One of the culprit is carbon dioxide. So the question is that whether bacteria can now take carbon dioxide to make fuel. So I'm quickly going now because of the time. So there is a technology, there is a science actually calling it solar fuel, solar chemical technology. We are calling liquid sunlight. you can actually liquefy the sunlight. When sunlight is available, you can store as an energy, not like it's all electricity, in the form of liquid fuel. That means in chemistry we are calling bonding. So if we keep as a bond, you can keep it is in the form of energy. So the thing is that at the moment we are using a linear economy. That means we are using petroleum-based fuel, which is making a linear economy. Linear economy means we are releasing a pollution like CO2. So we need to close this loop, okay, to make circular economy. This is a recent trend that we are actually moving to a circular economy. So my research is to close this loop using bacteria. So how we can actually use bacteria to convert carbon dioxide to fuel, okay? So I will show how we are actually doing the research. Not working. So we know, right, sunlight. Sunlight is one of the abundant renewable energy source. Theoretically, if we can store 1 hour sunlight, you can actually satisfy 1 year energy demand. So if you have 1 hour sunlight, that is enough to run 1 year energy demand. Unfortunately, we cannot store sunlight perfectly because we have only solar panel, which is only running when sunlight is available. We cannot store it. So the question is that whether we can store the sunlight. Then we found a technique using bacteria. Here again, bacteria is coming to action. Use bacteria, some light harvesting material, and feed the carbon dioxide. You can make chemical from carbon dioxide. For example, you can make fatty acid, fatty alcohol using carbon dioxide. So I will show some of the very interesting discovery which we made it using bacteria. So the speciality of bacteria is that they are living. That means you can regenerate. You can use for decades the bacteria. And in Cambridge, we made a special setup, a photo sheet. This is a photo sheet we made. It is made up of with the 2 unit in chemistry perspective. We have a water oxidation unit one side. We have a proton reduction unit in another side, which is really like a photosynthesis mechanism. And then we are actually grow the bacteria on this photo sheet. This is a photo sheet. We grow the bacteria. Here we grow the bacteria. And what we did that, we pass the sunlight through this one. Then using the sunlight, bacteria taking the electrons and eventually converting carbon dioxide to acetic acid. So acetic acid is a fatty acid. It is a precursor or raw chemical for many expensive chemical. So we are just feeding carbon dioxide to bacteria using sunlight, and bacteria converting carbon dioxide to acetic acid. Okay. Then whenever you are actually making anything, you need to prove it, right? So people will ask what is really happening. So we did some experiment using isotopes. Maybe in +2 or in degree you learn what is isotopes. So isotope actually giving a mechanism, what is really happening inside the cell body. And using NMR technique, there is a spectroscopy called NMR, you can actually track the mechanism, what is really happening when during the mechanism. So this is called a Wood-Jundal pathway in the acetogenic bacteria, and eventually we are getting a very good amount of acetic acid. And actually I got interviewed for this phenomenon. Now we are actually making the large scale. So now it is just a lab scale. So now we are making this one 100 meters square, which is actually very, very large, and you can actually making a liter of acetic acid directly by feeding carbon dioxide. Then finally, this is my final thing, what is a very recent one, which is accepted 2 weeks before. This is one of my hot topic, which I am actually doing. So dividing the labor between 2 microbes. So normally, even human beings, we can't do any job alone, right? We need a different expertise. So in nature, bacteria actually helping each other. That is, we are called division of labor. So one bacteria doing one job, another bacteria doing another job, helping each other, making some phenomenal changes. So here what we are doing that, we use 2 special microbes. One is my favorite bacteria, Geobacter sulfur reducent. Another Archaea, which is making methane, that is methanol barkery. There are two microbes coming together, helping each other by dividing the labor. You can see, I'm not going the details, but the concept is that we are dividing the labor between two microbes to do a very fancy types reaction. So here what is happening that we are using this conductive nanowire and connecting with another bacteria. You can see the plug-in mechanism. It is plug-in, helping. What is really happening that they are both are happy to do a job. So if we can selectively pick up some microbes and put in the laboratory, you can do whatever job you want. So we can actually control all the microbes to do whatever job we can do. So here we did a good job to make methane from CO2. We can do many jobs. As I said, we can use butyric acid, caproic acid, even aviation fuel, which is we are using in the aeroplane. So using bacteria. So bacteria, some bacteria is not specialized, so we are coming together. And so this concept is very popular, microbial division of labor. So how we can divide the labor. So we can see that we did lots of proof, because when you are actually saying something, you need to show multiple evidences to prove that one. So we showed lots of evidence what we are really doing, how it is happening. So I'm not showing that one. So in summary, if somebody asks in my research what I did from my PhD to till now, so I developed very highly performing fuel cell reactor using bacteria. That means I can actually produce electricity from waste using bacteria, which was my PhD thesis. Then I used the electric syntrophy to produce electricity from plastic waste. And I used the urine-powered microbial fuel cell, which we can run the bulb or, as I said, LED bulb, even the mobile battery charging using urine-powered microbial fuel cell. Then use a photo sheet to produce chemical from sunlight, carbon dioxide and bacteria. And eventually, we divide the labor between two microbes to do some very sophisticated job using microbes. So normally, people ask you how we are doing research. So normally, we need a very high funding to do the research. For example, if you want to do genome sequence, gene expression analysis, you need to pay just for a simple analysis £7,000. That means just a 1 hour job, we need 7 lakh Indian money to perform a single experiment. So we got lots of funding. Before that, he is my Marie Curie advisor from University of Cambridge. And these are my colleagues when I used the photo sheet experiment. She moved to Japan now. She is my current collaborator to use spectroscopy, because there is a question still we need to address. How bacteria taking electron from catalyst? This is unknown now. So she is helping to track the mechanism using spectroscopy. These two people gave us so many funding. The Johnson Matthey is one of the very big company in UK. They are actually interested to make hydrogen from waste. So they gave me a funding to support a PhD student, Martin Hayes, from Procter & Gamble. I know, maybe you know the company Procter & Gamble is one of the biggest company in the world. So they also supported for the thing and got. So these are all the funding agency from UK. So this is one of the biggest funding agency, Royal Society. And I was part of this Marie Curie Association, and we got a big funding from Research England. So the building which I made using urine-powered, that building, we got from funding from Research England. We got 8 million funding from this Research England. So finally, this is my final slide. And I'm very thankful to this bacteria, because my life actually changed using this bacteria. And they appeared in big journals as a cover image. This is a chemistry of material. This is our bacteria, and using covered by graphene. Chemistry of material is one of the leading journals from American Chemical Society, and it's from Royal Society. This is our bacteria making water splitting reaction. You can see the bubbles are coming because of oxygen and hydrogen. And it's our JACS, Journal of American Chemical Society. It's the Bible of chemistry. So we published a cover image here. So we got lots of cover images as a highlight of our research. Anyhow, this is my research last decade, since 2013 to till now, and many thanks. And sorry if it is dragging your attention. Thank you so much.
Thank you so much, sir. We are deeply grateful for the enlightening presentation and really encouraged by your research journey and also very much inspired by your experimental biological house. We truly appreciate the time and effort you took to share your knowledge with us.